with 1 being the maximum value that it could take in terms of its contour length. We said that the data actually looked a little bit like this. I'm just drawing this wrong, but it should asymptote to this value here. And we tried out various fits. One fit was just assuming that this was an ordinary Gaussian polymer. The second was assuming this was a freely jointed chain. And the third was the calculation that we did the last time, which was the semi-flexible polymer. So remember that for this, the relationship was force that had to be applied was thrice kBt divided by n L squared times r. And so r is the same as L times x, I guess. You could use that. And the second relationship that we had, so this was for the Gaussian chain, the ordinary polymer that we started off discussing earlier. Then for the freely jointed chain, we had a result that said that the extension, let us say r here, was n times L into 1 minus 1 over, and I had a factor of kBt divided by f times L, where L was the basic size of the step. So this, as I made f large, this quantity would go to NL. So that's what this divergence is like here. It comes from here. So it goes up like that. The third one was a worm-like chain. So if we just write that out, worm-like chain, otherwise known as the semi-flexible polymer. And this took the form R equal to approximately n times the persistence length Lp into 1 minus, so sorry, n times L here, into 1 minus 1 over square root of 4 times beta times f times the persistence length Lp. Okay? So this is the 1 over square root of S behavior that I pointed out was important, and this gives you a fit that goes much closer to the actual data. Now, to go back a little bit to what this description meant, remember that we started off by saying we wanted to look at a polymer that was like this. This was our DNA molecule, and we said that we should think of this as being a kind of cylinder, and then ask what are the sort of deformation that you might apply to a molecule in the shape of a cylinder. We said one thing you could do would be to pull at it from this side by exerting a certain force per unit area, and then there would be some extension delta L, and we said, or we showed that F divided by A, which we call the stress, was proportional to, I think I used the quantity, what notation did I use for this, Young's modulus? The E was E times delta L divided by L. The other things I said we could do was to say that, look, take your cylinder and imagine that I draw a line here, always parallel, an imaginary line here, always parallel to some axis, say the z-axis, and then I turn this around like this, so that now this turned in this manner. The vector here moved around here, and because this is an elastic medium, over here it would start turning around and would reach its maximum value here at this particular point. So this I called a twist, and a measure of the twist is what is the angle that it makes here. If this angle is 0 here and I impose an angle phi here, what is the energy cost of that deformation? The third one was I took a cylinder here and I bent it across like this. And if I sort of drew this line like this, one way of doing it would be to ensure that this line that fits through the center of the cylinder is a constant radius. It's part of a bigger circle with some radius r. So this corresponds to inducing some curvature in this. So there would be some cost for curving this particular cylinder. And I said that this was the important one that we would try to model. The fact that it costs energy to bend the molecule as opposed to pulling it and stretching it, and as opposed to twisting it. So if these energies are high, I can assume that the molecule is not pulled, it's not twisted, but it can be bent because it's the smallest energy in the system. And typically the energy associated with curving this or bending it is typically comparable to the temperature, to kBT. So then we asked, how would we do this? So what we said was, let us imagine a normal way of thinking about a polymer says, take a coordinate system, draw your polymer here, and write the vector r as a function of some quantity s on that polymer. So this might be s equal to 0, and this might be s equal to L, and s anywhere in between associated with some r. Okay? From here, from this r, we can get a tangent vector t hat, which is dr of s by ds. And if this is a unit vector, this has to be divided by the modulus dr of s by ds evaluated at the same s. So this is now a unit tangent vector. So remember what this is doing. Here is your r. So this might be r of s. This would be r of s plus ds. And the tangent vector is something that points between these 2 points, between r at s and r plus ds, and it is normalized so that it has unit value. Okay? So now, if it is normalized, and if I integrate between 0 and L of d t hat of s by ds times ds, this should give me just L, because this quantity is 1, and I'm integrating over s. Okay? So this is a constraint to be incorporated, and the constraint that mod t is defined to be equal to 1 is a constraint that says that there is no pulling involved. This I can deform it, but I have to deform it at fixed overall length. Okay? And that is how I impose this particular condition. Having done this, remember that we said we can write the energy of the system as some quantity Lp divided by 2, an integral over this internal coordinate between 0 and L times d t hat of s by ds the whole squared. This is saying that first of all, if t hat of s is a constant, then all these vectors point in the same direction, and there is no energy cost at all, because t hat remains the same irrespective of wherever each of these vectors points in the same direction. It's only if they don't point in the same direction that this tangent here might look like this, might be here, this might be here, this might be here, this might be here, and so on. Only then does this pick up a value here, non-zero value. Because of the square, whether I deform it like that or I deform it like this, which are both physically completely equivalent things that I can do to a structureless polymer, the energy of this and the energy of this is the same, and this comes only from curvature. Remember that t was defined as dr by ds, so dt by ds is d2r by ds squared. It's a second derivative, okay, which is the curvature of how this thing is bent across. Something that is straight but at an angle has a first derivative, but the second derivative vanishes. So curvature is associated with the second derivative. The fact that the tangent itself to the curve must change from point to point as you move along. Let's just think a little bit about the dimensions here. This has, so this should be E, I think this should be kBT. kBT times Lp divided by 2 is the value that I think I should have. No, there's already kBT hidden inside the Lp, so I don't need it there. Yeah. All right. So this is the form that it actually has to take. And from here, is that correct? Have I made a mistake there? I think it's E divided by kBT here. That is Lp by 2 times that particular form there. We can go back and check that this is right a little later. So I wanted to do just one simple calculation, and that's a calculation that takes something that is straight and finds the curvature energy involved in taking it and making a circle out of it. Okay? So here's an example of something that has curvature. I've taken it and made these two ends meet here to form a round loop in the form of a circle. And now I want to ask, what are the energetics of this particular system here? Now, I can define my S variable here to be the same as or equivalent to the variable theta. So, ds here is actually r times d theta, because a small distance between here and here in terms of theta translates into an arc length of r times d theta. And theta must now vary between 0 and 2 pi. That's the amount that it is supposed to vary. So now how do we think of this vector t hat? So t hat is a unit vector, t hat at some s, which is t hat as a function of essentially the single variable theta over here, is a vector that points in this direction always. So this is the distance from the radius to that point. The t hat variable, the tangent variable, points in the theta hat direction by when you use spherical coordinates, or in this case simple polar coordinates. So remember what polar coordinates are. I can either take a point and describe it in terms of some point here in terms of x and y, or I can say let me describe it in terms of a vector here in the direction r hat. So how far I have to go in the radial direction from a point, and how far do I have to go in this direction, in the theta hat direction here. So any vector field in 2 dimensions has some component, suppose at some point r comma theta. So I parameterize this point by r comma theta. So any point on the plane I can describe by what is the r value it has, what is the theta value it has. And any vector in the plane has a component that is directed along the unit vector along r and perpendicular to it along theta hat. Okay? So now you can see, just sort of looking at this, that the vector t hat here is essentially just the same as theta hat, because that's a vector that is always tangent to this particular curve here. And it's a unit vector by creation, and therefore it must point in the theta hat direction. So now when I look at d t hat of s by ds, this is the same as d theta hat by ds, which is the same as d theta hat by d theta times d theta by ds, which now d theta hat by d theta is the same as minus r hat, and the reason is just geometrical. Suppose here I have a vector that is in the theta direction, and over here I have a vector that is in that direction here. The difference between this is obtained by taking this here, putting it here, and this is the difference which points in the minus r hat direction. Okay? So when I do that, I have d theta by ds is 1 by r. Okay? Therefore, this particular quantity here, this energy here divided by kBt, is just Lp divided by 2 into the factor of this particular thing here, 1 over r squared, r hat dot r hat, because it's a squared here, that gives me 1, and I must integrate between 0 and 2 pi of ds, which is r d theta. Let me write all of those terms out neatly and in a helpful way. The terms that I will have are E over kBt is just Lp divided by 2 into 1 over r squared into r into 2 pi, which is just pi. 2 and 2 go away, r squared gives me r. So pi times Lp divided by r. So this is the energy. Okay? So notice that as I make my arc become bigger and bigger and bigger, as I go to larger and larger circles, the energy associated with the curvature becomes smaller and smaller. And that makes sense, because the larger and larger I make it, the smaller is the deviation between adjacent tangent vectors. In the limit that r goes to infinity, it's really as though at any different point, at any point on the circle, it looks as though it's locally straight. So there's no energy cost associated with that. And that's how these results come. So this is just intended to get you to thinking about how to do this calculation. The difficulty with doing this is the fact that at every point, we must ensure that t hat equal to 1. So because by choosing t hat equal to theta hat here, we explicitly put that in and did all the calculations in 2 dimensions. But in general, remember what we have to do. The general calculation is calculating a partition function, which is exponential of minus that particular term here, LP divided by 2 integral 0 to L d t hat s by ds squared, the whole squared times ds. And what comes here is a complicated thing. What comes here is the restriction that you must sum over all configurations of the t hat of s vector. That means a sum over all shapes. Yeah. How does? So this is d theta hat by d theta. So that says that suppose I'm here at this particular value of theta, and my vector points in this direction. As I make theta going to theta plus d theta, that vector points in this direction. So the difference between these 2 vectors is this vector minus this vector, which points in this direction here. Now this direction is a direction along this, along the radial side, which means it is proportional to r hat. Okay, and minus sign because it's pointing inwards. The r hat normally points outwards. That doesn't need to be a unit vector. The derivative need not be a unit. The vector itself must be a unit vector. Okay.
And we know that r is a function of s, right? And we're integrating over s. So this s is the curvature, isn't it?
S is not the curvature. S is some parameter that tells me where I am along the chain. So I have a chain like this, and I have some way of measuring a unit, some internal dimension along this. So if I start here at s equal to 0, then I must end here at s equal to L. So the integral of ds between 0 and L must equal L itself. That's the total length. So in a sense, what S measures is, in the integral over S, measures the contour length of the polymer. Okay. All right, so this is just a quick calculation meant to sort of remind you about how to calculate the difficulties of calculating with t hat of S. And the fact I mentioned something the last time, that if I took, in principle, if I calculated this, and then I evaluated t hat of S dot t hat of S prime, and I took the average of that, this quantity behaved as exponential of minus S minus S prime divided by the persistence length, which is why the persistence length was important. So let us just plot it, and I should have plotted it the last time. So this is S minus S prime mod. Okay. So at S minus S prime equal to 0, which is at the same point here, the correlation is 1. But now the curve decays as you move away, and it decays as exponential of minus mod S minus S prime divided by LP. The larger LP is, the slower the decay is. So this is LP large, and this is LP small. If LP was infinite, then this would essentially stay stuck at 1 throughout. So LP measures how stiff the rod is. So the rod stiffness. The larger the persistence length, the stiffer the rod is, and the more difficult it is to change its configuration. The smaller LP is, the easier it is even for thermal fluctuations to disorder it. And the rough idea is that LP is that distance along the chain at which the energy to deform it becomes of order kBt. So, if I have I should think of my semi-flexible polymer in some, let us say, solution at finite temperature. So here is my polymer here, and as sort of molecules strike it from here to here, its configuration changes from here to here to here, so on and so forth. Okay? So now, question is how much does it change? And one way to do it is just to look at the correlation of the tangent vector here with the tangent vector at some other point. So this is t hat of s prime, this is t hat of s. If the chain is long enough, what is s and s prime doesn't matter. If this chain goes off to some very large here and very large here, this particular quantity depends only upon s minus s prime, the difference between s and s prime. Okay. So now this says that as I move further and further along from the chain, as these 2 points are more and more separated out, they can fluctuate more independently of each other. So if I'm at distances much larger than the persistence length, essentially these are uncorrelated with each other. They can fluctuate independently. So LP, the interpretation of LP is, it is that distance along the chain at which the fluctuations, the energy cost of bending it at that length scale becomes of order kBt. Okay, that means even thermal fluctuations are enough to seriously disorder the chain. The larger the LP, the stiffer it is, and the weak, and the further and further and further you have to go in order to make it deformable by thermal fluctuations. So that's the interpretation of LP, which I call the persistence length. And we might come across this persistence length again. For DNA, the persistence length is about 50 base pairs or 150 nanometers, if I remember. I think that's right, yeah. Or is it the other way around? I think it's just, I think this is the right way of saying it. And I'll have to check this. I don't remember what this value is. Okay. All right, so now we are done with polymers, and let us now do something else, which has to do with membranes. So both polymers and membranes are examples of extended structures inside the cell. The cell is full of things that are not just simple small molecules, but are fairly large and structured in some way or the other. So the polymer itself is a long molecule. Remember that the total DNA in any of your nuclei is around 2 meters in length, distributed over 23 pairs of chromosomes. And membranes, everything in the cell is practically enclosed in a membrane. The cell membrane itself separates the inside of the cell from the outside of the cell. So that sits here. There are many organelles. For example, the nucleus itself is surrounded by a double membrane here. Mitochondria are membrane enclosed. Vesicles are membrane enclosed inside here. Most organelles within the cell have some sort of membrane covering that protects them. Yeah.
The nucleus is covered by double membrane.
Yeah.
And cell
The cell is covered by a bilayer membrane. Here the nucleus has a double bilayer with a space in between.
2 layers are there.
Yeah. Okay. So now what we would like to do is to think a little bit about what is the role of these structures here, what determines their energy, determines their free energy, and what sort of shapes they can take. So the basic constituent of these is something called an amphiphilic molecule. Amphiphilic, the word amphiphilic means loving both. So it's a molecule that typically has a head group and a tail group, can either have a single tail or double tail. And the loving both refers to the fact that the head tends to be polar and hydrophilic for that reason. Whereas the tail groups tend to be non-polar, hydrocarbon tails, and hydrophobic, which means they don't like water. So the combination of the sort of molecule combines into some part that likes water and some part that dislikes water. And it's this amphiphilic character. For example, soaps are amphiphils because there are soaps usually sit at the interface of oil and water, with the heads pointing towards the water and the tails pointing towards the oil. Okay, so the tails are not exposed to the water at all. So it's these molecules that form the essential building blocks of cellular membranes. These are called phospholipid molecules. And that's because the tails are attached to the polar head by a phosphate group, a PO4 group that sits over there. These tails can be something like 8 to 12 carbon atoms inside. So the total height of this might be something like 2 nanometers, whereas the cell's bilayer membrane, which looks a little bit like this as a schematic, has a dimension which is about 4 to 5 nanometers.
Sir, let me ask one thing. This is a single layer, right? Is it a bilayer?
This is a bilayer. A single layer would be something that looks like so. This is called a lipid bilayer. The reason it forms a bilayer is there is water both on the inside and the outside, which is why the head would like to be exposed to the water on both sides. If I had a water-oil mixture, this might like to sit at the interface between water here and oil here. So these structures would be formed under such circumstances. So over here, there are 15 to 18 carbon atoms in these hydrocarbon tails. These can be single bonds, or they can also be double bonds. And double bonds induce a kink in the tail that expands the region that is required for the tail. So here are a couple of different arrangements of amphiphilic molecules. One arrangement, of course, is just the homogeneous arrangement, where I have molecules that float around independent of each other at sufficiently low densities. If the density is low enough, this is always a preferred state, the homogeneous state. As I increase the density, these can begin to organize in interesting ways. One way in which they can organize is to form something that looks like this. This is called a micelle, okay, where all the tails are packed in together and exposing the head to the outside. Another structure that you can have is a structure that looks like this is called an inverted micelle. Yet one more is called a cylindrical phase. And in the cylindrical phase, what you have is something that looks like this, where all the heads are exposed to the outside, and maybe I could even put a bilayer in this, looking with heads both on the inside and the outside. And this whole structure here forms a cylinder. And the last one, which will be the most relevant to our purposes, is the bilayer membrane. And as I said, the cell membrane that encloses the cell in eukaryotic system is a bilayer membrane, which is structured to prevent the inside and the outside from communicating, from the same of protecting the chemical environment of the inside from the environment of the outside. Exactly which of these states are preferred depends upon the dimensions of the molecule. For example, if the molecule looked a little bit like this, if it had a large head group compared to its tails, it would tend to form a micellar structure more easily, because then it's just by a packing consideration that would enable you to pack these cones together more sharply. If, on the other hand, we had the head group and the tails being approximately the same size, the symmetry of a cylinder here, it might be easier to pack into some other configuration. Of course, the other extreme limit would be where I had a small head and a large tail. So now I would have something that looked like a cone in the other direction, on the other side. So now let's do a simple calculation of when membranes begin to aggregate, or when aggregates begin to form from the homogeneous state. So let's pose the question as, when do aggregates form from the homogeneous state? And the answer is that there is typically a concentration that need not be very sharply prescribed, but it's an approximate separation of the homogeneous and inhomogeneous region. This is called the critical micellar concentration, or the CMC. So what this means is the following. I take a solution, I put in some low density of amphiphilic molecules. If the density is low enough, they all remain independent of each other and floating around. And you expect this because the entropy is much more if you allow them to float around and not communicate much to each other. In order to force them to form some structure, you must basically make it energetically favorable for it to do so. There must be some energy gain from doing so, from forming some sort of ordered structure. The way to find out where this critical micellar concentration that separates a homogeneous state from the inhomogeneous state where things begin to pack together is to compare the effects of entropy with the effects of the energy term. That's essentially what one has to do. So let's just go back to an old calculation that we did already, which is in order to calculate the entropy. And our approach will be, we will calculate the free energy, which is the internal energy U minus T times S, and find out what are the properties of the free energy. When does it go from being energy dominated to being entropy dominated? When does it change sign? Okay? In order to do this, let us just assume that this homogeneous state, where I have some low density of these molecules moving around, This is like the ideal gas.
So, but they both have to be like inverse of each other, right?
Hmm?
If energy is, if we are increasing the energy for it to form micelles,
Yeah.
the entropy has to minimize, right?
Not necessarily. I mean, it turns out that it will have to, but so we will do a simple calculation that tells you how to balance one against the other. And how increasing the energy by reducing the entropy of this. So this is a high entropy state because I allow all of these to wander around independently. So I reduce the entropy of that by confining some of them. In order to make sure that that state that I get finally has a favorable free energy, I must compensate the reduction in entropy by some increase in binding energy. And that is what this particular term contributes. So let us just try calculating the S first. And so to do this, let me go back to the old calculation of the ideal gas. Remember that the ideal gas, if I just think of this as a gas of molecule, these don't interact with each other, so their only energy is potential energy. So this is 1/2 p squared divided by m, p squared divided by 2m. From this, remember that we calculated the partition function for n such particles, which is just the integral over all the momenta e to the power minus beta p squared over 2m for all of these particles together. Remember that we wrote the answer for z as v to the power n divided by n factorial lambda to the power 3n. This is the form that we finally wrote it in. And the expression for lambda that we had lambda is h squared over square root of 2 pi m kb t. And this, as I remember, I have done this already in somewhere in your notes for just the calculation of the partition function of the free particle. Do we have this? Do you remember this?
We have done till here, p square by 2m.
And then I calculated this. This is just a Gaussian integral, which I remember doing. And then I wrote it in this final form here, this very simple form, which just involved lambda. Okay? So from here, remember that F, the free energy, is minus kBt times the log of the partition function, which was minus kBt times the log of v to the power n divided by n factorial lambda to the power 3n. What is? H is the Planck's constant. So once I did this, I used Stirling's approximation. I wrote this first as minus kBt times log of v to the power n minus log of n factorial minus 3n log of lambda. I just took the log and put the minus signs correctly. Then I said that this is minus kBt times n log v, and then I used Stirling's approximation for log n factorial. And that just says the log of n factorial is n log n minus n plus higher order terms. So if n is large enough, I just need to worry about keeping these two terms. If I do that, I get n log v minus n log n plus n minus 3n log of lambda. Now I can pull n out and write this as minus n kBt, and then I have a log v and a log n here. So I have a log of v divided by n here, and then I have a term that looks like plus n plus 1, this should be because I pulled the n out, minus 3 log of lambda. And this becomes v divided by n is inverse density. It's the number of particles per unit volume is n divided by v. So I can write this as the free energy here. Let me worry about all my minus signs carefully. If I take this minus sign inside here, so let me first write that term. This is minus n kBT times the log of V divided by n plus 1 minus 3 log lambda. Correct? So now I can take the minus sign inside and write this as n times kBT times minus log V by n minus 1 plus 3 log lambda. Okay? Now V divided by n, so now log of V divided by n is log of 1 by rho, which is minus log of rho. So this is Stirling's approximation. So this is something that you have to read up because it's too long to explain here. So this is Stirling's approximation. And this is just a way of approximating for n large enough what is log n factorial. And the answer is that this is given very well by n log n minus n, and other order terms are also of order 1 by n and higher order. So if n is large enough, those are the two terms that dominate. Okay. So this term here is minus log rho, so this becomes n times kBT times log of rho. In fact, I can write this term as log of rho lambda cubed minus 1. So this is the free energy that we have here. So now let us calculate the energy gain from putting these molecules together. So let us say that that energy gain comes from an energy of binding these molecules together, which I will call E binding, which is the binding energy. And I want to make an argument for what this binding energy is. And I'm going to say that this energy really comes from the effective area of the hydrocarbon chain into the water hydrocarbon surface energy. So what this is saying is that when my molecules are free to move around, they're exposing these hydrocarbon tails to the water. By forcing them to come together in some structure that looks like this, they are now reduced that interaction with the water, because now the hydrocarbon tails don't have to intersect water anymore. So one way of doing it is to say how much area is this exposing to the water, and multiply that by the energy required for an interface between a hydrocarbon and the water tail here, which they can approximate in various ways. All right. So now if I imagine that the molecule itself forms a structure that looks like this, each of these, this would be Nc, which is the number of carbon atoms, into LCc, which is the carbon-carbon bond length, and rho is the number density. Yeah. Okay. So if this is the radius r of the hydrocarbon, so I have a structure that looks like this. The binding energy is just 2 pi times R, hydrocarbon, times Nc times LCC times gamma, where gamma is the surface tension. Okay?
So how did you get lambda cube? Is it using
I got it from here because 3 log lambda is log of lambda cubed. Okay, so now we are in a position to calculate the free energy together. And this is the energy gain from binding. So remember that the free energy is the internal energy minus T times S. Okay? So if S dominates, then the free energy is negative, because this I can ignore and this becomes, S is always a number that is greater than 1, greater than 0. So this will always become very negative if S is large. Okay? On the other hand, if this quantity dominates, then this thing will be positive and this will be small. If the entropy is small, then this term will win out. So these two you can roughly balance by saying where the energy is of order T times the entropy, that is where the transition between things being homogeneous and moving around versus the collapsed condensed state that we drew out here. This is where the transition actually happens. So now let's begin to equate this and say that okay, we have to do one more step from here. So this is the free energy over here associated with the molecule. Now the internal energy E associated with that. So this is E bind of order Ts is what we have to calculate. The energy associated with that, with the homogeneous state, is just 3 by 2 N kBT, and therefore the entropy is the energy minus free energy minus the average energy divided by T. I think I've got a sign right. Let me just check that. F is E minus Ts. So Ts is E minus F. Yeah, my mistake. This is average energy minus F divided by T is the entropy that I have. Okay? So then I have a factor of from here, I have S equal to, the T and T goes away, so I have 3 by 2 N kB. And then I have my other term here. And if I get rid of this T, this becomes minus N times kB times log of rho lambda cubed minus 1. So this is now my entropy S associated with the homogeneous state. Is it clear what I did? I went back, I said, all right, I have now calculated my free energy here. From my free energy, I can calculate my average energy in many ways. Simple way to do it is to take a derivative with respect to beta and put a minus sign, where beta is 1 over kBT. We worked this out earlier. The other way is just to remember that this is something that is particles with just a completely quadratic Hamiltonian, because the energy is just a sum of quadratic terms, 1 over 2m p squared. So 1 over 2m p1 squared plus p2 squared plus p3 squared plus p4 squared. Automatically I know that when that happens, each of those quadratic terms can be assigned an energy of kBT. Okay? So there are n particles, there is px, py and pz. So each of those quadratic terms, p squared is px squared plus py squared plus pz squared for each particle, n particles, 3 directions. So that is thrice n kBT divided by 2. Okay? That's exactly the logic that is. So that's why I was able to write it down immediately in this particular form. Once I have done that, I know my free energy has a form of energy minus T times S. I just calculated this first part. So from there I can get the entropy, which is this. So this is the entropy of the state in which the particles are free to move. They are homogeneous. I must now compare this with the energy gain of putting the particles together, which is my binding energy. So let's do that. So first let's just simplify this term here and write S of the gas state or the homogeneous state as 3 by 2 n kb plus n kb, which is 5 by 2 n kb, and then the other term, which is minus n kb times log of rho times lambda cubed. Okay, so that's the gas entropy here. And now I must equate that to E binding. So T times S gas is this object times T, and I must set this equal to E binding. So what I will get is,
So we know that is gas.
one second, let me just check this. What we will get is rho. So let me just write these two terms out here. I can pull the kb, so let me just write out this term first here. This becomes 5 by 2 n minus n log rho lambda cubed times kb t equal to E binding. So E binding I lose a factor of n. Let me just write this first. E binding minus 5 by 2 n kBt, by taking this term here and putting it there, is n log rho lambda cubed. And I have to get rid of that factor of n somehow. So I'm just wondering whether the way I should get rid of it is to just write this E binding here as epsilon times n, where this is energy per molecule into n. So if I do that, I have epsilon times n minus 5 by 2 n kBt is n log rho lambda cubed, in which case I can get rid of all of these factors of n. So if I do that, what I will get is rho times lambda cubed is the exponential of epsilon binding minus 5 by 2 kBt. Okay. And provided I have my signs right.
So minus sign, minus n log rho lambda cubed.
So I should have a minus sign here, a minus sign here,
Minus a, yeah, minus there.
minus there. Okay, that makes complete sense. So then I have minus and plus, I think. Somewhere I need a factor of KBT as well. KBT also. Okay, perfect. All right. And then I have a KBT factor here. No, I have a KBT factor here. Okay, perfect. That's exactly what I wanted. I have a factor of E binding divided by KBT, and the second term is just 5 by 2. Okay? So what this says is that at the critical micellar concentration, rho becomes rho star, which is the density rho star, that satisfies this particular relationship between the binding energy and the density here. Okay? We wrote the binding energy in terms of some quantity that I have now rubbed out, but let me just write it down again. So E binding is 2 pi NC R HC LCC times gamma. So what this says is that if I plot here, if I plot the rho star, which is the critical micellar concentration, I take a log on both sides, which means that I have to plot this on a log scale. And the scale that I will choose starts off at 10th power minus 10, 10th power minus 8, 10th power minus 6, 10th power minus 4, 10th power minus 2. And I plot this, then this quantity becomes log of this is just this quantity here. This quantity depends upon NC. So I plot NC here. What I should get is a straight line with a negative slope. Huh? The gamma is the surface tension. This has to be an energy. So what this says is, if I'm protecting, if I just let this float around like this here, it's exposing these tails to the water. Okay? These tails don't like water. So there is a certain energy cost involved in exposing them to the water, which I will call gamma. Okay? When I pack them together, I save this energy. I don't have to pay it anymore. So then the question is, how much energy have I actually saved? So that energy is estimated by saying that I take these molecules together, there is so much of it that is exposed that's proportional to this particular height, it's proportional to the number of carbon atoms inside this. So the height is the number of carbon atoms times the distance between each of those carbon atoms. And then I need an additional factor, which is what is the dimension of this part, which is the RCC, RHC. Once I do that, it's this sort of rectangular structure that I make from here that I now have freed from paying for this energy, for the interfacial energy. Once I do that, I put these NC's factors in. Then I can basically find this particular relationship between the CMC and NC. So the longer NC is the number of carbons in the tail. By doing appropriate biochemistry, I can change the size of the tail from something like this to something like this. If the energy is really an energy that comes from the exposure of those tails to the environment, then the simple calculation of what is that energy gain from protecting it will tell me that this energy has to be proportional to the number of carbon atoms, which gives it the length of the cylinder. Okay? Once I put that in, because essentially that enters in this factor here, and this is the relation that tells me that this should be my density at which I now begin to aggregate these molecules together, then this predicts a relationship between the number of carbon atoms in the tail and the critical micellar concentration. And this is a very strong dependence because this is a exponential dependence on the number. This E binding depends upon NC. So this is exponential of that, which is usually a much more stronger dependence.
So what is that?
NC is the number of carbon atoms in the tail. RHC is the the radius of this, essentially it's the radius of this here, the structure here. So I'm just approximating it by cylinder.
And LCC is the height.
LCC is the distance between adjacent carbon atoms.
Adjacent or
Yeah. So if I multiply LCC by NC by the number of carbon atoms, that gives me a measure of how long the molecule is. So this is the exponential dependence of the critical micellar concentration on the number of carbon atoms in the tail. Okay. So now we have thought a little bit about what these molecules look like, how their behavior is influenced by the fact that they are amphiphilic, that they are composed of both hydrophilic parts, which is the head group, and hydrophobic part, which is the tail group, therefore making them amphiphilic. How this property of the molecule, as well as the shape of the molecule, influences the way these molecules pack, and thereby influences what are the structures that they might form. Okay. For structures where the tails can be packed well and tightly together, where the head group and tail group don't differ too much in size, the structure that is preferred is the structure of the bilayer. And now we have to try and understand how do we describe the bilayer inside a cell, and how do we understand its properties. First of all, notice that there are many lipids, or many different types of lipids, which are part of the cell membrane. In addition, there are also sterol molecules, such as cholesterol, which enter here, which are small molecules that also sit here and give the cell bilayer a certain rigidity. In addition to this, there can be large membrane spanning protein molecules which also sit on the surface of the membrane. So these are called transmembrane proteins, which cross from the outside to the inside. In addition, there could also be more complicated complexes of proteins. For example, you could have ion channels that allow ions to go from one to the other. You can have water channels that allow water to pass from one side to the other. The whole membrane, the actual cell membrane, is an extremely complicated object. So now we have to try and figure out. Huh? They are They're also they're also composed of proteins that line this. Yeah. The other thing to notice is that the lipid molecules on the membrane are in a fluid state under physiological conditions, which means that if I look at a patch of the membrane, and over here are lipid molecules in the bilayer here. If I look at where a particular lipid molecule is at a given instant of time, if I look at it again, it can have diffused quite far away from where it was originally. That means the environment is not tightly constrained. These are much more like liquids, where molecules can flow past each other, and not like solids, where they have a fixed relationship with each other. So these are called fluid membranes, and all of our discussion will be focused on fluid membranes. And one example that is particularly useful to study is the red blood cell, which is not really a cell because it doesn't have a nucleus. It's essentially just crammed full of hemoglobin, but the covering of the cell is pretty much like a regular fluid membrane without very serious complications from all of these additional accessory proteins that the usual cell membrane has. So that's a good model system to actually study. So what we want to do is to think a little bit about how one must describe fluid membranes, what is the mathematics that one must use in order to do that. So let's just do one quick description of the way we will describe the molecule, and then we'll take it up again the next time. The way I will describe the molecule is to say that, look, I have a Cartesian coordinate system here. Here is my membrane, which is some sort of surface that sits here. I will specify my membrane by saying, given any point x comma y, there is a height h of x comma y that tells me where the membrane is. Okay. Now, in order to specify a surface in 3 dimensions, I need to specify 2 coordinates to you. I will choose these 2 coordinates in this particular simplified case to be the x and y coordinates, and the membrane to be the z coordinate, the height of the membrane to be the z coordinate of the point. So then the r, which tells me where the membrane is given by x comma y comma h of x comma y. That's how I will describe the membrane. This is sometimes called the Monge representation, or the Monge representation of a fluid membrane. And we'll describe this in some more detail the next time, what it does and how to begin calculating some very simple things and how to find the analog of the persistence length in this particular problem and to ask what it means. So, h is the height above the xy plane. So the xy plane is z equal to 0. I can specify a point, a surface by saying for any point in the xy there is an associated height. If h of xy is independent of x and y, that's a flat membrane altogether. And the reason I must have a coordinate system that is outside this membrane is because all of these, it cannot be tied to each of these molecules themselves, because the molecules are free to move around and diffuse around. So if I want to talk about the shape of the membrane, I must say here is my coordinate system, and here is where any point on that surface is sitting. So these membranes are often also thought of as random surfaces. That's one good way of thinking about them. It is still 2-dimensional because we're sort of assuming that we won't worry about the thickness of the membrane. That's only some 4 to 5 nanometers in thickness. But we're interested in quantities that are much larger than that thickness. So in that approximation, this is just an infinitely thin sheet. But I must account for the fact that it's a sheet. And we'll describe this a little later the next time.